Welcome to The Explainer. You know, the title of this 1984 medical paper, Unidentified Curved Bacilli, doesn't exactly sound like a page-turner. But the discovery inside, well, it completely shattered a core medical belief and totally revolutionized how we treat one of the most common stomach problems in the world.
So, what really causes stomach ulcers? For most of modern medical history, we thought we knew the answer. It seemed obvious, it made sense, and, as it turns out, it was almost completely wrong.
Yeah, for decades, the story was always the same. If you had that burning, gnawing pain of a peptic ulcer, the blame was put on your stressful job. Maybe that's spicy food you ate, or just your body making too much stock acid. It was just this chronic lifestyle thing you had to manage, not something you could ever truly cure.
But then, this paper comes along. Written by two Australian researchers, Barry Marshall and Robin Warren, it pointed the finger at a totally new culprit. They proposed that all that pain, the inflammation, the ulcers themselves, they weren't caused by your lifestyle at all, but by a tiny, microscopic invader.
Okay, so let's dive into this. Honestly, the best way to understand this paper is to treat it like a detective story. So we're going to start by opening up the decades-old cold case file on peptic ulcers. You see, the problem with the whole acid and stress theory was that the story just didn't add up. As the paper itself points out, you could use powerful drugs to block acid and heal an ulcer, sure, but the underlying inflammation, the gastritis, it was still there. And almost every single time, that ulcer would come roaring back. The treatment was really just a Band-Aid.
And just like in any good mystery, the crucial clues had actually been there for years, just sitting in old lab reports and medical journals. They'd just been, well, overlooked or dismissed as totally unimportant.
Get this. As far back as 1940, researchers spotted these weird spiral-shaped bacteria in stomach tissue, but the findings just got forgotten. Then it happens again in 1975. Another team sees similar bacteria in a huge majority of their ulcer patients, but they just figured it was probably contamination from their equipment. The prime suspect was seen at the crime scene multiple times, but nobody ever identified them.
So enter our detectives. It took these two persistent researchers from Perth to finally connect the dots. Warren, he was a pathologist, and he kept seeing these strange bacteria in inflamed stomach biopsies. And Marshall, a young gastroenterology fellow, actually took him seriously. They decided it was time to open a formal investigation.
So what did they do? Let's get into their investigation. It wasn't anything flashy, but wow, it was meticulous. They set up a study to look at 100 patients in a row who were coming in for a routine gastroscopy. The goal was simple. Look for this mystery bacteria and see if there was a real connection to the disease.
Okay, so here's how they did it. It was actually super straightforward. During each procedure, they'd take two tiny tissue samples from the patient's stomach. One sample went to pathology to be checked under a microscope for inflammation, what's called gastritis. The other one went to the microbiology lab, with one simple instruction: try to grow whatever is living in there.
And this is where the story gets really good. This is the lucky break every detective dreams of. At first, the lab couldn't grow a thing. They were throwing the culture plates out after the standard 2 days. But then, a total accident. A set of plates got left in the incubator over a long Easter holiday weekend. When the technician came back, they found growth. Turns out, this bacteria was just a really slow grower, and they'd been tossing it out before it ever had a chance to show up.
So now they could finally grow the bacteria in the lab. This meant Marshall and Warren could finally analyze all the data from their 100 patients. They had the pathology reports, and now they had the microbiology reports. And when they put them side by side, wow, the results were absolutely astonishing. This was their smoking gun.
I mean, just look at how strong this connection is. For the patients who had the bacteria, pretty much all of them, over 96%, also had gastritis. Now look at the group without the bacteria. Almost none of them had any inflammation at all. The link was it was undeniable.
And it wasn't just gastritis. The correlation with actual ulcers was just as strong. Of the 31 patients in the study who had an ulcer, 29 of them, that's the vast majority, were also infected with this bacteria. This was no coincidence.
You know, the authors themselves pointed out how wild this was. PMNs, they're a type of white blood cell, kind of like the first responders that rush in to fight an infection. To see them swarming all around these bacteria, right there in the stomach lining, well, they argued that was incredibly strong evidence. In their words, the bacteria couldn't just be some innocent bystander. It had to be causing the disease.
All right, so they've got their suspect at the scene of the crime and they've tied it directly to the damage. What's the next step in any investigation? You've got to formally identify your culprit. You've got to put a face to the name.
So using a powerful electron microscope, they got the first clear mug shot of their culprit. And you can see it right here, this curved S-shaped rod just nestled right on the surface of the stomach cells. You can even see those little hair-like things it uses to move around. Those are its flagella.
So they put together a full profile of their suspect. It was a new species they initially called pyloric Campylobacter. Later it'd be renamed Helicobacter pylori. They noted its unique set of 4 flagella and realized just how perfectly it was adapted to survive. See, it lives in the mucus layer that coats the stomach, which is almost neutral. This protects it from the super harsh, deadly acid just millimeters away. Pretty clever, right?
So, let's talk about how this discovery didn't just solve a mystery. It completely shattered a miracle dogma and changed the lives of millions and millions of people. This is how the case was closed.
I mean, you have to understand, the implications were just staggering. This one single finding transformed peptic ulcer disease. It went from being this chronic, incurable thing you manage with a bland diet and lifelong meds to what it actually was. An infectious disease that you could permanently cure with a simple, short course of antibiotics.
And with this one single powerful line from their paper, Marshall and Warren just threw down the gauntlet. Diathesis is basically a medical term for being prone to something. They were suggesting that the entire idea that some people were just ulcer-prone because of stress or their genes was probably a myth. The problem wasn't the person, it was the infection.
The story of H. pylori is just such a powerful reminder that science is never really settled. It took 2 skeptical researchers, a little bit of luck, and data that you just couldn't argue with to overturn decades of established medical thinking. And it really leaves you wondering, right? What other medical certainties of today are just waiting for their own Easter holiday weekend in the lab?